On to Dr. Weeks. Dr. Weeks will be presenting on bullfrogs in Colorado. As far as the Western Slope, she will be talking about her citizen science work and how bullfrogs have affected West Slope amphibians. So welcome, Dr. Weeks. We're super happy to have her here. She drove out from Grand Junction and might be one of the further attendees we have for this meeting. So thank you for coming out.
Thank you for having me. I'm really excited to be here and share this with you. So I figured it might be helpful to give you a little bit of an idea about what inspired this whole Bullfrogs for Beer citizen science effort. So I got to Grand Junction, in case those of you in the audience don't know where it's at. It's on the western side of the state over there. I got to Grand Junction in 2018, and since 2019 we've been working to cooperative agreement with multiple agencies to try to catalog the diversity of amphibians in this canyon system that we didn't know much about. So if you look here, I'll show a few maps that'll kind of show in different ways. But in the far left says the MCNCA, so the McInnes Canyon National Conservation Area. Pretty remote. As you get deep out in that canyon, pretty much like hiking down from the top 10 plus miles or boating in and going up. So we didn't know a lot about the amphibians that live there, So this project, this is actually showing that McInnes Canyon area, or Black Ridge Canyons Wilderness it's also called. So that yellow area has a whole bunch of canyon systems in it. And the Colorado River, you can see right there north of that whole section, flows through that. And this is about a 24-mile stretch that is commonly recreated, so rafters use this a lot. And the priorities with this project that we started on were to catalog the diversity through visual encounter surveys, but also look for where bullfrogs are, because we'd kind of gotten anecdotal evidence that they existed there, but we weren't sure where. So we were doing visual encounter surveys with those, but also collecting water samples to do eDNA. And then every amphibian that we captured, we were swabbing so that we could look for the presence of BD. So since then, it's really cool. This cooperative agreement has supported 8 CMU students to do these research projects. So I hire students every summer and they get to go out and have this experience. And I don't have an acknowledgement slide at the end, but as I go throughout, I'll talk about different people that I've worked with. So for the cooperative agreement, I primarily worked with David Pilliot at USGS, Nikki Grant Hoffman for BLM, and then Dan Newbaum with CPW. So we've been kind of the main players on this. We go out and sometimes we have to boat, sometimes we hike long distances, we hike out in these canyons. These are just a couple of the students who've had this experience. And then USGS has been working on the GPS data that we collect to make maps of where these guys are at. So coming back to show you this one more time, here's just one of our pictures of us boating down the river. And some of these students have never been on a boat before, so this is a really fun excursion for a first research outing. When we go to conduct these surveys, we go out at night to do call surveys and visual encounter surveys. Again, we swab the ones that we capture. And we go out during the day too, mostly for our water samples for that eDNA. Any individual that we capture, we are collecting our GPS data for. We describe the habitat. Here's a couple more photos showing the type of habitat we have to clamber through. So on the right, we're climbing a waterfall. So it's kind of hard to access to get in there in some of these spots. And the 5 species that we have out there that we've been looking for, we've got woodhouses, our Great Basin spadefoots, our red-spotted toads, our canyon tree frogs, and the northern leopard frogs. All 5 of these have been found out in these canyon systems. We weren't sure, with the northern leopard frogs, we have not found them up in the canyons, but closer to the Colorado River. And as I mentioned, we were collecting the eDNA, the water samples for the bullfrog. So I want to point your attention to this middle picture here where it shows that kind of slabby rock environment with some water there. So the majority of these canyons look like this. So the water will be there from maybe late April through early June, and then by hot summer it dries up. So with this being the kind of habitat that we see, not a lot of vegetation, we've found all different toad species breeding in here, canyon tree frogs, spadefoots, and the concern is our bullfrogs making it up in here too. All right. So I'll zoom in on this map in a moment, in the next slide, but this map kind of showing you where it is. And really importantly, I'll point out that here's Grand Junction. And then we have over here is Palisade and Fruta, so a couple of towns we have in the valley. And the Colorado River flows to the south of these towns, and then the north over the McInnes Canyon area. And you'll notice there are a couple of little pluses there with these frogs in Grand Junction and Fruta, and all the way out there McInnes Canyon. This is a map that my student put together for a presentation where we had found bullfrogs at parks in the towns and also out in the canyons. Those positives represent where we found BD positive samples on bullfrogs. And that was from 2019. So if I zoom in, this is just a map that we're still trying to update the map. This is all the way back to 2020. But I'll give you an idea, a rundown of what we found. So each one of those colors represents a different canyon. And the Devil's Canyon, Pollock area over here is just pretty easy to hike into. The rest of these are really long hikes or you have to boat in. So the really important things I'll point you to, we've found canyon tree frogs in almost all the canyons. We found spadefoots and both red-spotted and Woodhouse's toads. But the BD is that yellow box there. We actually found that at the mouth of Mead Canyon, so right where it meets the river. We found that on a bullfrog. And the one above that that's in turquoise, eDNA, or Leica, which is Lithobates cataspinus, so our bullfrog. So for eDNA for that, we also found it at the mouth of Mead Canyon by the river. And you'll notice in that top portion, East Salt Creek, that area is one that does not dry up. It's a very different habitat. It's a really important one used for fish breeding as well, that CPW, I think, works on some species that use that waterway. So bullfrogs are high density in that region. And then as you go up, there's one more, VES for LICA, it's that like pink one. And so we found those again at the Mead Canyon mouth. They're in East Salt Creek. And this last year we found them at the edge of Rattlesnake. So again, by the river. Up to this point, we had thought, well, I showed you some of those waterfall pictures. Bullfrogs can't climb very well. So we thought maybe these geographic barriers would keep them from moving up into the canyons at these higher elevations. And that was what the data might have suggested up to this point. This past summer, we found them 2 miles into Mead Canyon, that just changed the whole story. So now we're shifting efforts to maybe some more bullfrog eradication in these canyons, doing a couple hikes out there. This is what some of those waterfalls look like. So we couldn't imagine that a bullfrog would get up over this, but they're either coming down from the top or they're finding a way up from the bottom, maybe by skirting some of these waterfall habitats. So where are the bullfrogs coming from? As we're collecting this data, we're finding that they are down there, and they're at the bottom of these canyons, and they might move up soon. Maybe they're coming from these source populations in Grand Junction and Fruta and Palisade, where we have these parks and these ponds that are right next to the river, and they're just teeming with bullfrogs. And bullfrogs, as we know, are our carriers for disease, and they also eat anything that moves, including our native species. So these are a few of the parks that my students and I honed in on initially. And of those 3 parks, the 2 on the left were the ones that we found BD positive bullfrogs in. So we think the Colorado River might be a corridor to bring these bullfrogs downstream to these canyon habitats. So Bullfrogs for Beers was born because I thought it takes a small army to get people to come out and help remove bullfrogs. And what's a greater motivator than beer, right? If you're not as passionate about catching bullfrogs as I am, bring beer into the picture. So I thought this might be a fun thing to do, get the community involved. I worked closely with CPW on the marketing materials for the event because it's something that's a bullfrog eradication event. We want to be clear with the public what that message is and make sure they understand why we're moving them and what's going to happen to them. Volunteers were recruited through advertisements. We have a local math and science center, so I did a lot of trainings there, and they did recruitment for people there that were interested in conservation. Also Facebook, emails, interested groups, so we have Audubon Society, a chapter over there. River's Edge West is a conservation group over there, so they would kind of send an email out to all of their groups that are interested. And on the bottom, these are all the organizations that help bring this together. So as they mentioned earlier, CoPark was really helpful in giving us some grant money to buy some of those rewards for people to come out. Base Camp Beer Works, they were the first brewery to jump on board. They used to be called Monumental. They've been out there recently. They are really interested in the river conservation, so they were just like, yeah, anything that helps conservation, and they just gave us a bunch of free beer coupons, which is awesome. The Eureka Math Science Center, River's Edge West, and CPW. So the trainings, over Zoom, some people preferred, so I would just do a few Zoom meetings in the summer, and they had to do a training before they came out to collect, because I wanted to make sure that message was clear. Why are we collecting these bullfrogs? How are we going to go about it so it's safe and humane for the animals? And all these rules for collection events. So the objectives here, the community education piece was really important. Getting them involved in removing an invasive species, understanding why they are invasive there. And then the BD testing and diet analysis, this created new research projects for students at CMU. So the students do all of this part. They do the diet analysis. They're also doing the BD testing from start to finish. They learn that skill. And then, ideally, I have a lot of bullfrogs in a freezer right now. I would like to donate them to a local high school to use for dissection labs, so they don't have to buy these that come from a bullfrog farm somewhere. And we just haven't gotten all the way to that piece yet. I've made some connections, but we haven't actually physically moved the bullfrogs over. So some of the training slides, just a couple of them, what I would tell people when they get on this Zoom call, that we're collecting some data, including the profile of the bullfrogs that exist in these parks, the disease status of the bullfrogs. We're going to look at their gut contents to see what they're eating. And at the event, I would tell them to expect that everyone is required to complete a virtual training, so if they weren't on my list, they didn't get to participate. They would sign a liability waiver at check-in, just in case they fall in the pond and get hurt. They can't get mad at me. We would then do a walk over to the pond, and I would help discuss with them exactly what we're sampling and kind of how to move about the habitat. I would give them gloves and teach them that they're supposed to change their gloves in between each animal so that I can test for BD. So they were trained on the whole disease transfer protocol. They'd bring them over, give me their dirty gloves, and get a new pair. And that check-in table where I would have the gloves, and I would have a few of my students helping me. We would do masks and snout vent length, we would do some BD swabs, and then we would put them into an individual baggie, into a bucket to take back to the university. And initially, it was 2 bullfrogs get you 1 beer voucher, but some people were so good at catching bullfrogs that I had to bump it up a little bit this past summer. And I forgot to mention that we didn't just have beer vouchers, right? Little kids would come out sometimes too, and sometimes the parents would bribe them into giving them their beer vouchers. But I had a local dairy, our local ice cream stand that we were able to use some of the grant funds to purchase some little ice cream vouchers that were about the same value. And we also had a local pizza place hop on board this past summer. So we're starting to grow in the rewards that we have to offer to people. And one of the things they're also trained on was, of course, how to identify a bullfrog. So we're not just grabbing leopard frogs and stressing out other frogs that are in the pond. Because all of these ponds where we were capturing bullfrogs, we have seen leopard frogs there. They share this habitat. So only bullfrogs can be captured. I'd explain how to identify them. I would tell them no gigging of frogs, because this is going to look bad, right? If people are gigging frogs and bringing them to me, somebody else might be horrified. So they had to come to me alive and in good condition. They could do hand captures, what I encourage, because I think that's more fun and it's a little more like you build a skill there. But also live capture nets. They were given these capture nets that they could cinch closed once they captured an organism. But I had cleaning bins at the booth. So if they did capture one, they would have to come up, and it was a bleach solution that we know kills BD, and they would have to do that cleaning before they went back out. Of course, had to give them this one to unattended children and let them know that there's a scientific collection permit that's kind of an umbrella permit that covers them only for those events. But that if they have a fishing license, that they would be allowed to capture them outside of collection events at a later date. And a couple of important things about this that I decided with CPW was we didn't have more than 12 people in an event, and this was initially because we did this for the first 2 years to see how this was going to go and make sure that do I have enough people that I can manage? Because I'm running this data table, but I also need to make sure that I can watch the volunteers and everyone's being safe and treating the habitat and the animals appropriately. So the collection locations have been the Snooks Bottom one in Fruita. It's a big open space, it has fish in there. This Connected Lakes or Audubon Nature Preserve, they're kind of two parks that are connected into one. And then Corn Lake was one that we added this year. Since then, so we've done this for the last 2 years, I already mentioned some of those businesses that have partnered, as well as some local conservation groups that have jumped on board. We've had 81 volunteers total. We've removed 91 bullfrogs. Oops. And here's just one of the data tables, and then in the background you can see that one is the nature preserve area, one of the collection sites. So I also want to mention here that the nets were donated by River's Edge West, these live capture nets that were especially helpful for little kids that came out. And then the funding for the prizes and other kind of supplies we needed at the event, both donated between the Co-Park grant and with this brewery. So the 2021 events, we had 3 training dates over Zoom that summer, and then we had an event on July 30th where we caught 27 bullfrogs, August 2nd, Snooks Bottom, where we caught 16, and August 3rd at Snooks Bottom, we only caught 5, right? And this picture in the middle is a couple of volunteers already out there at dusk trying to lay out their strategy. In case you're interested, I have some information about all the bullfrogs that was captured. This top picture is nice because it shows you from an aerial view where the Colorado River runs along these parks and why I chose these. So you can see the Colorado River there, looks like that brown color coming through, and on the left, there's a series of ponds there, connected lakes. And we chose one of those kind of up toward the top that we worked in, but this is just a stone's throw from the river, and when the river does flood, it connects with these water bodies. We didn't have any VD at this park in that year. So the first year that I detected VD here was 2019, and I haven't detected it since. 2021 at Snooks Bottom, we had 14 females that we caught and 7 males. And so this one, again, you can see there's the Colorado River in the middle, and then right there on the left, that big kind of squiggly shape, that's Snooks Bottom open space, right there next to the river. Also, we didn't find any VD this year, in 2021. In 2022 events, it just keeps growing, and I hope to expand this this coming summer. We had 3 training dates, again, over Zoom. July 9th, Snooks Bottom again, we caught 10 bullfrogs. You remember the first one was, I think, it was a total over 20 bullfrogs at that one. We did a connected lake sampling, we caught 19. And we also had a Girl Scout troop reach out to me and take part in this. So we did one for them on August 18th. And you see here in the bottom, this is me leading a training. I decided that on-site training might be better for a Girl Scout troop than trying to maintain their engagement over a Zoom call. So I brought little packets with, and they learned about why this was important and how to identify all the different frog species. Some of them were really knowledgeable too, so it's awesome. We caught 14 bullfrogs that night. So to break it down for each night, the Snooks Bottom one from this last summer, we caught the 10 bullfrogs, all of them were large adults, 4 females, 6 males. We're still working on those BD samples. I still have some students that are working through some of this lab work to identify what those results are going to be. For Connected Lakes, for the Audubon, 19 bullfrogs, there's 14 juveniles here, 2 females and 3 males. And I forgot to mention it on the first year of collection for Connected Lakes. Almost all of them were also juveniles. Over 20 of them were juveniles, very few adult males and females. Also, BD samples are pending. And then this was the night with the Girl Scouts, 21 volunteers, but this was because we had both parents coming out with the kiddos. This is a pretty big pond, so Corn Lake was one we chose for this one. So you can see the Colorado River there, and then right above that is that big Corn Lake open space. And here we caught 6 females, 4 males, and 4 juveniles. Still working on those BD results. But I do have some interesting results for you about what they were eating. And the interesting thing about this is that in 2021, we have gone through all their stomach contents and we have not found vertebrates. but from 2022. You can see this frog in the top left. You can see how big he is compared to those diet contents. We actually found, this is a Woodhouse's toad. It's a juvenile Woodhouse's toad that it had eaten. And then the top, I know it's kind of hard to tell, but it's a fish. I couldn't identify what kind of fish. It was so far digested. But you could see the little, the fish bones and some of the fish skin left over. And then on the bottom, this one took some investigating. When you look at the bones in this closely, we figured out it was a pelvis of an anurin, but we don't know if it was a frog or a toad yet. We're trying to figure out based on some ratios of the bones if we can narrow that down. But so the first frog ate a couple of vertebrates, the second one, pelvis of an anurin, and then this one from Corn Lake had eaten a juvenile garter snake. So you can see kind of the size comparison there. This is significant because we're trying to work toward, or at least I'm trying to apply for a grant that would expand this kind of citizen science that we can do over Western Colorado. And we have evidence now showing that they are actually eating our vertebrate species, including amphibians, including snakes. What I'd be really interested in to see is the ponds that we collected from, that we saw this in, these were ponds with plenty of insect diversity. But remember that landscape that I showed you in the canyons, right? It's not very, there's not a lot of vegetation. There's kind of this slabby rock that dries up really quickly. So most of the items moving around them are probably going to be other frogs in those environments. And I'd really like to get into those canyons and start getting more diet analysis on the bullfrogs that are in there and see if they're eating some of our sensitive amphibian species. Future directions for this, I'd like to include more locations and more events and expand partnerships with other breweries. As you might imagine, any kind of government funds cannot be used to directly buy beer as a reward. So that's something that I've run into. But people have suggested other ways that I might be able to redeem that. And if anybody here has suggestions, I'm open to hearing them. Because that seems to be a fun piece of this, and we have a lot of breweries popping up in Western Colorado that want to be a part of this. And then involving river rafters. That was kind of my original dream for this. We know these bullfrogs are on the river, and people are out there recreating. We have an annual river cleanup day where they just do a camping trip, that 24-mile distance, all the way down. And it would be awesome if we could set up these kind of weekend events where we have like pods of people that go out and they camp by where these bullfrogs are living, and then we have collection events there. So that is the dream, ultimately. The people that I've been working with in CPW on this, Lori Martin and Albert Romero, have been really helpful in helping me develop this and supporting this project. With that, I will take any questions that you guys have. I'll start there and I'll go to you next.
If you set a bunch of bullfrogs in a freezer that that you don't have anything to do with, maybe you can invite people to help catch the bullfrogs to a grill or something like that.
To a grill. So I forgot to mention that the way that I euthanize them is with MS-222. So probably won't do a grill with those. Yeah, but we overdose them with a fish anesthetic just so that it's kind of quick and painless for them. And somebody had a question back over there.
Yeah, I was wondering if you're seeing them more associated with permanent bodies of water, not so much with transients, temporary areas.
So these ponds that we pulled from for the citizen science, these are permanent bodies of water, and definitely that was an assumption that I had. But then we found about 7 bullfrogs 2 miles up into one of those canyons that is ephemeral and it dries up within midsummer, usually every year.
We looked at them in the southeast extensively a long time ago, and they're really greatly associated with permanent ponds in the areas that are transient, open bay, but then during dry years, knocks them back.
Yeah.
You have a dominance in native species.
And what I have noticed is 2019 was, I guess, what I would call a good water year. And this past summer was too. And the years where we have a lot of rain and the water is lasting longer in those canyon habitats, we see the bullfrogs out longer and they move up into the canyons more on those years.
We were wondering how they get in these areas around waterfalls. They can disperse during rain nights really long distances, so they can go around trucks.
Yeah. I wondered if they haven't been washed down from the top too. We have some higher elevation areas, but there's still like some farmland up there people have, and they might wash down from upstream. Yeah.
Do you have any sort of population monitoring to just measure the effects of the removal, or are you just relying on your removal numbers?
So initially, I just wanted to see if this would work, if people would be interested, and this would like be a good thing. And this last summer, my colleague Paul Hampton was like, well, why don't we start doing some kind of monitoring before and after an event to see if it makes a difference. And the type of approach that we had chosen was doing a call survey, because we were already kind of going out so much and we didn't have a lot of people helping with surveys. And you start to realize that just those males calling isn't going to give you that picture of the females that were removed and the juveniles that were removed. So I think this next summer we're going to try to actually go in and do some like deeper surveying, going around in the pond and looking for the individuals. Yeah.
Is there any data out there about how successful different frog and tail species are in coexisting with bullfrogs? You mentioned in one place that there was both leopard frogs and bullfrogs. I mean, is that usually just a one-way ticket to living for the leopard frogs?
I don't know of any data that I can speak of, but I know that leopard frogs and bullfrogs coexisting. There's some evidence of bullfrogs eating leopard frogs. Leopard frogs are very susceptible to BD, which bullfrogs can carry, and I think it's kind of we start seeing these leopard frog declines in the data over the years, and one of the hypotheses is that bullfrogs coming in started to lead to those declines that we see in leopard frogs, at least where I'm at. Yeah.
So is there any information as you go along the river there through that 24 miles, that there are sites along that river that could serve as your source sites as well? So, yeah.
I think so, because when you get down to the drainage part that meets the river, you start seeing that pond-like habitat. You've got cattails, you have a really like perfect environment that looks like a pond. And they're hanging out in Mee Canyon for sure, and this one looks a lot more like this pond-like habitat toward the exit. So I do think that there are some that we could focus on. The nice thing with me trying to get river raptors involved is these are campsites also, so people can camp there and it'd be really easy to do removal.
How can you like sign up to like go catch bullfrogs and stuff?
I can, when I have some events planned for this year, I can maybe send an email out to these guys and they can let everybody know when to come out. That'd be awesome, yeah. Any other questions? Yeah.
Did you guys, what do you have? Did you collect any data on native herbs that you saw during this?
Yes, thank you. So that map that I showed you that had like the, I'm gonna go back to really quickly. I have a lot of animations. I'm almost there, I think. So what I wanted to mention about this. So here, we just made an assay for the eDNA stuff that we collected we have 3 years of data. And initially we just started off looking at bullfrogs, but now we're making assays to look for leopard frogs as well. So with that same water sample, we can start seeing maybe we just didn't see the leopard frogs, but they were there. And this information is part of a federal database now, but we're working on a paper, so this information will be out within the next year, hopefully. And I plan to put a website up so that people who are doing the citizen science stuff can come back and see what, their efforts resulted in.
Got one last, well, probably be your last question. Utah is just over the border. You can see it there on the line. Are there any implications further downstream in collaboration with Moab and so on and so forth?
So there's this group called CCAST, and they're like a collaborative toolbox for aquatic invasive species removal. And there are some groups that I've seen come on there that they're working on their own bullfrog removal over in Moab, so they do have issues with it there as well. Yeah.
Hunter, harness all the spring break energies you can see as you.
Come down and just
It's so
remove
cold
them.
out
Yes,
there.
Moab would have a whole other frame of reference on
It's still cold, man.
college students.
We're talking about it. We're all in the back saying, "Oh, bullfrog opportunities, let's go!" So I think we'll be out there. Thank you, Dr. Weeks. Appreciate you.